Hello friends. Now let us start with the portion of biotechnology, microbial biotechnology. In this first we'll in short discuss about the history of the first chapter, that is recombinant DNA technology. A scientist called Herbert Boyer, who was born in 1936 in Pennsylvania, a small country, and he graduated from the University of Pittsburgh, followed by Yale University. He was actually an assistant professor in the University of California at San Francisco. By 1969, he performed studies on restriction enzymes that are found in E. coli organism. He observed that these enzymes were able to cut the DNA strands into a particular way, leaving the sticky ends at the end of these strands. On these clipped ends, pasting of pieces of other DNAs was possible. During the same time, one more scientist named Cohen, a Stanford scientist, had been studying on the small ringlets of DNA that are called as plasmids, which are the autonomously replicating circular extrachromosomal materials found in the bacterial protoplasm that we know very well. Cohen had developed a method of removing these plasmids from one cell and again reinserting them in the other cells of bacteria. So Boyer and Stanley Cohen thereafter worked together on combining their processes of DNA splicing on plasmids, which enabled them to produce successfully recombinant segments of plasmid DNA of desired configurations, and they inserted them in the other bacterial cells. Such bacterial cells with this recombined DNA could then act as factories for production of specific proteins. This breakthrough was the actually basis upon which the discipline of biotechnology is founded. Biotechnology deals with the techniques of using live organisms or their enzymes to produce products and processes that are useful for humankind or mankind. Thus, making curd, bakery bread, or wine, which are all microbe-related mediated fermentation processes, could also be considered as a form of biotechnology. However, term biotechnology is used in a restricted sense nowadays to refer to these processes which actually use genetically modified organisms. Further, many other processes or techniques are also included under biotechnology. For example, in vitro fertilization leading to a test tube baby, synthesizing a gene and using it, developing DNA vaccine, or correcting defective genes, etc. are the different techniques that are performed under biotechnology nowadays. Thus, the European Federation of Biotechnology, that is EFB, has given a definition of biotechnology as the integration of natural science and organisms. The integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services. I will discuss about the principles of biotechnology. The two most important core techniques that gave birth to modern biotechnology are genetic engineering and microbial chemical bioprocessing. Technique of alteration, genetic engineering is the technique of alteration in the chemistry of genetic material, DNA or RNA, and its introduction into the host organisms, thus changing the phenotype of the host organisms. Microbial bioprocessing is the maintenance of sterile condition, that is the microbial contamination free environment in microbial processes to enable the growth of only the desired microbe or eukaryotic cells in large quantities for the manufacture of biotechnological products like antibiotics, vaccines, enzymes, etc. Students, you know very well the advantages of sexual reproduction over the asexual reproduction. The sexual reproduction provides opportunities for variations and formulation of new combinations of genetic setup, some of which may become beneficial to that organism or may be useful to the industry. Asexual reproduction, however, preserves the genetic information for generations together in the individual. While traditional hybridization procedures are employed, especially in breeding of the animals and plants, which often come with the inclusion of and multiplication of undesirable genes along with the desired genes. However, modern microbial biotechnology is the answer to the problems which are seen in these sexual, asexual reproduction or hybridization procedures. These are the techniques of genetic engineering, which include creation of recombinant DNA, use of gene cloning and gene transfer so as to overcome the limitations and allowing us to isolate and introduce only one or a set of desirable genes without introduction of undesirable genes into the target organisms. Imagine what will happen to a piece of DNA if somehow we introduced it into a new organism's protoplasm. Most likely, this piece of DNA which we have introduced will not multiply itself in the cells of that organism. But when the unknown piece of DNA becomes the part of chromosome that has ability to replicate, it will surely be inherited and multiply into the progenies. This is because in any chromosome there is a specific DNA sequence called as origin of replication, which is responsible for initiating replication. Therefore, for the multiplication of any piece of DNA in an organism, it needs to be the part of chromosome, which has a specific sequence in it, that is origin of replication. Thus, after integration of new DNA, it is linked with the origin of replication also. So, that piece of DNA now can replicate and multiply itself in the host organism. This can also be called as cloning or making multiple identical copies of any template of DNA. Let us now understand how to make an artificial recombinant DNA. Previously, we have seen the work of Boyer and Cohen. The construction of first recombinant DNA emerged from their work. They were working on possibility of linking a gene encoding antibiotic resistance with the plasmid of Salmonella organism, that is Salmonella typhimurium. Stanley Cohen and Herbert Boyer accomplished this work in 1972. By isolating the antibiotic resistance gene by cutting out a piece of DNA from a plasmid which was responsible for conferring antibiotic resistance. They employed the enzymes for cutting DNA strands and characterized those enzymes. The cutting of DNA at a specific locations became possible with the discovery of these enzymes, so called as molecular scissors or the restriction enzymes. The small cut pieces of DNA were then linked with cut sticky end of plasmid DNA. Such modified plasmids were introduced into protoplasm of bacteria, making them antibiotic resistant. Thus, such plasmid DNA were employed to act as vectors to transfer the piece of novel DNA attached to it. You know well that what is vector. The antibiotic resistance gene with the plasmid vector genome was performed with the help of enzyme that was called as DNA ligase, which acts on cut ends of DNA molecule and joins their ends. This makes a new combination of circular DNA